So in this lesson, we're going to follow up on your Meiosis 1 and 2 worksheets and point out some details that are worth pointing out, which are the key importances of meiosis. So in other words, what makes meiosis different from mitosis in ways that are really important to recognize? So let's start. Well, the first one you're probably thinking is fairly obvious. You've probably guessed it, which is that number one, it produces haploid cells. And why do we care about the cells being produced by meiosis being haploid? Well, that's because in order for the sperm coming from one parent and the egg coming from the other, they must combine to have a full set, in other words, a diploid number of chromosomes. No more and no less, right? So if sperm and if egg were not haploid in number, then sperm plus egg would not equal the diploid number of chromosomes, which is the standard set of chromosomes in all body cells, with the exception of the reproductive cells. So it's super, super important. Now, the second point I'm hoping you're able to see from your study of meiosis 1, which is that it produces genetically different cells. Whereas mitosis will produce two daughter cells that are genetically identical to the parent. In other words, mitosis, you start with a parent cell and then you produce two daughter cells that are genetically identical. So in other words, if this is a skin cell that needs to reproduce, you're going to end up with two genetically identical skin cells resulting. Now, this is, in ideal circumstances. Of course, there are factors that can affect that, but the intention of mitosis is to produce genetically identical cells. Whereas if you have meiosis, one of the goals is, so for example, you'll have a gamete cell that's produced, that's going to be produced that is different than the other three gametes that are also produced. And why is that? Well, if you have any siblings, then you know that your siblings definitely look like you, but they're not identical to you, unless, of course, they're your twin. In the same fashion, you have probably you bear some resemblance to each of your parents, but possibly to different extents. And that's a function of the fact that, yes, indeed, your gametes are coming from your parent cells, but there's also been some genetic modification from your parent cells along the line. So during meiosis, there are two distinct happenings that ensure this genetic difference that exists when gametes are produced. So let's take a look at that. So the first one is, the first event is what we call crossover, or chiasma is the German, or the Latin name for that. And so you'll remember that this happens during, try to think of the phase where this happens. Well, it happens during prophase 1. So, phase 1 of meiosis 1. So, what's going on here? Well, you probably remember from either your reading or the video that was suggested that you will have your homologous chromosomes aligned during prophase 1, and what will happen is that you will see the chromosomes in that tetrad. So, tetrad meaning 1, 2, 3, 4 chromatids. That will cross over and then exchange genetic information. So this is an event that does not happen during mitosis, during your regular cells being multiplied. This only happens during meiosis. So let's say for instance you have your mom's chromosome and your dad's chromosome in that homologous pair. Well, they're going to exchange information, and when they exchange information, each chromosome in that homologous pair moves on to the next phase, which would be metaphase 1, and they would have separate different genetic information than they would beforehand. And so even here we can see that, you know, when they separate during anaphase, when their centromeres are split, they are going to be going off separate, but you can see that each of these chromatids is unique. Okay, so that means that the 4 haploid cells that are going to be produced by meiosis are going to be also genetically unique. So there's an example of why you are going to look like one or both parents to different extents. And also why maybe you look like your siblings, but maybe aren't identical to them. So the second event that ensures genetic variation in gametes is what we refer to as independent assortment. So this is a difficult concept to explain, and so I'm going to do my best to try and explain it to you. So we would say that in a homologous pair, chromosomes from the mother will move towards one pole and the father to the opposite pole. So, you know, I'm going to break this little part in half here and say, okay, well, you know, that's certainly true. I know that if I have in a homologous pair here, I know that I have a mom chromosome and a dad chromosome in that set, and I know that, you know, eventually, you know, they're going to end up in a different cell, and this at the end of meiosis 1. Right? So for instance, maybe this guy here and this guy here, right? And then go on to divide again, and so on and so forth, right? So the fact that they segregate from each other ensures genetic diversity. But that's not really what we mean by independent assortment. You'll remember that even though I've only indicated one pair, we need to remember that that's going to exist times 23, because we have 23 pairs of chromosomes per cell. So to simplify things, I'm going to try to explain this to you by showing 2 sets. Okay, so we know, for instance, here that I'm going to just draw an oblong cell. So I might have, let's say, like a big red chromosome and a big blue chromosome. That would represent one pair. And then somewhere in the karyotype, I might see a small chromosome and red chromosome and a small blue one, right? So again, you know, in each homologous pair, here's one homologous pair, here's a different homologous pair. One of these chromosomes belongs to one parent and the other one belongs to the other. So for instance, mom and dad. And here's another one, mom and dad. Well, we know that they're going to separate, right? We know that they're going to separate into different cells eventually. But the thing is that there are lots of possibilities of how that could happen, right? We know we're going to end up with 2 different cells, but maybe we have the case where, you know, the big red goes with the little red. Maybe we have the case where the big blue goes with the little blue. But it's also possible for instead to have big red go with little blue. That would be another possible combination in terms of the way that they could sort themselves out. And by the same token, I might end up with big blue and little red. So these are, you know, the 2 possibilities. I could either end up with this situation, and that's how they sort themselves, or I could end up with that situation. And you can imagine that I'm only showing you 2 pairs of chromosomes here, but we need to remember that that's going to happen for each of the 23 pairs of chromosomes that can happen. Let me show you another picture that will depict that. So we would say that each homologous pair sorts independently of the 22 others. So here they're only showing single chromosomes, I think probably to simplify things, right? But we know that, you know, your parents' gamete and, you know, mom, maybe dad and mom coming together will give you the 2 sets. So here they're showing 1, 2, 3 homologous pairs, but we know that the way that these guys, when they go to opposite poles, when cell division happens, that the way that they do that can differ. For example, all three of the blue could go to one end, and then all three of the yellow to the other end. I could have, two blue, one yellow, and on and on and on and on. There are lots of different potential combinations of chromosomes that could be in each of the gametes, right? Lots of different possibilities, which is why, people will resemble their siblings, but not usually in an identical fashion, because we have, 23 pairs that can all sort themselves in different ways, producing a different number of genetic possibilities at each turn. So geneticists will often calculate the number of gamete combinations that can happen. And so they use a handy equation to represent that. And so we know that the number of gamete combinations that can occur, in other words, the number of possible different sex cells that you can have, will be equal to 2 to the power of n, where n represents the number of homologous pairs that are available in the organism. So if we know that for humans, well, what is the number of homologous pairs? Well, for humans, n is equal to 23, right? We know that that's the case. But, in other organisms, we would use a different value for n. So if, for instance, you have a question such as the following: how many possible gamete combinations could result from 23 homologous pairs sorting independently? Now, in my example, I only showed you 2 sorting independently, and in the second example, there were 3 sorting independently. So you can imagine that with 23, there's going to be a lot of possible outcomes there. So I would say G is equal to 2 to the power of n. So in the case of humans, it would be 2 to the power of 23. So I would go ahead and I would calculate that on my calculator using my exponent button. And I'm going to go ahead and do that. And so when I type that into my calculator, this is the magic number I get. I get 8338608. So that means I've got, over 8 million different possible gamete combinations. So there we have it. So we've talked about the importance of meiosis as a process. Number 1, it produces haploid cells. Number 2, it produces genetic variation by way of crossover and by independent assortment. So there are some questions that I'd like you to try from your textbook on page 181 here, and I'd like you to try those out just to solidify your own understanding of these concepts, because we will be having a skills check very shortly. We have one topic left to cover, which is genetic mutations, and once we've done that, then we should be ready to do a skills check. So look for that in probably the next week or so. As always, reach out with any questions that you might have, or any points of confusion, any points of clarification that need to happen, and I'll be happy to assist you.